Our next speaker, the topic merges beautifully, flows beautifully from this one, is Dr. Brice Gaudilliere. He is an assistant professor of anesthesiology, perioperative, and pain medicine. He's done really important work on cytometry by time of flight spectrometry and its relationship to immune system in various patient scenarios, including in the preterm clinical scenario. Please help me welcome Dr. Gaudilliere.
Well, thank you, Yasser. Thanks for the introduction. It's really a great honor to be invited as part of this very esteemed and accomplished group of speakers. It's a bit intimidating, to tell you the truth. I want to start with two announcements. First, I don't think there's any scary pictures in my talk, so please please remain seated. The second thing that I'd like to start with is by mentioning that for this very important and global health problem of preterm labor, there's currently no diagnostic test. And with this talk, I'd like to introduce the idea that the maternal immune system may hold important clues into the mechanisms of preterm birth, clues that may hopefully at some point form the basis to develop a diagnostic test. So as Gary mentioned, and as well as our keynote speaker, Michael Liu, preterm birth is a very multifactorial problem with many different etiologies, a very complex problem to study. We've heard there's environmental issues, socioeconomic factors. We're going to hear this afternoon with David Relman talk that there are microbiome-related issues and immunological issues. And so our lab, and it's a collaboration with Dr. Martin Anks and Nima Gaipur, who's here in the assistance, we focus on the immunology side of things. Why immunology? Well, think of pregnancy as a very finely tuned immune balance, which for 9 months will really prevent the maternal immune system from rejecting a hemiallogeneic fetus, chock full of paternal antigen. And so mammals have developed very sophisticated mechanisms of fetal-maternal tolerance that are initiated at the beginning of pregnancy and maintained throughout pregnancy to help this phenomenon happen. But then, at the end of pregnancy, there is a very interesting switch in the maternal immune state. And it's been now characterized as a pro-inflammatory state, not unlike what happens in transplant rejection. And this switch in the immunology of the maternal system is important to promote all the physiological changes that happen with labor and delivery. And you can imagine that if this switch happens too early, we might be in a situation of preterm birth. Now, most of what we understand from the immunology of pregnancy comes from really the study of the fetal-maternal interface, and we'll hear more from Virginia Wynn's talk later on about the placenta. And so at the level of the placenta and the decidua, this is where all the maternal immune cells, and there are very many of them, many different cell types, come into contact with the fetal tissue. And so there are really important immune regulatory mechanisms that participate in this fetal-maternal tolerance. By contrast, very little is known about what happens in the periphery. And this is what our group focuses on. There you go. What we're really trying to understand is what are the immunological fingerprints that we can detect in the peripheral immune system that reflect the ripple effect of local phenomena that happens at the fetal-maternal interface. And there's two reasons why we focus on the peripheral immune system. First of all, peripheral immune cells have this really unique properties of circulating and coming into contact with pretty much every organ in your body and perhaps maintaining a memory of different pathologies that may happen locally. And second, the peripheral immune system is accessible. So with a simple blood draw, you can have many different immune cells, as many as 2 million in 1 ml of blood, 2 or 3 million. And so therefore, when you think about translating this into a diagnostic test, it's a good way to start. So now there are many ways that one can measure inflammation and the state of the immune system, of the peripheral immune system. We've heard from Gary, you can measure cytokine profiles, you can measure transcriptomic changes in pooled immune cells. But the limitations with both of these approaches is that they don't have single cell resolution. And to steal a Barry Bear's analogy, it's a little bit as if you're measuring or trying to understand what people in San Francisco had for dinner last night by measuring the sewage line. So you really don't know, if you're measuring TNF-alpha, for example, you don't know which immune cell has secreted TNF-alpha. Actually, it could be an endothelial cell. Nonetheless, they're really powerful tools and powerful methods, but to really understand the biology, you have to go at the single cell level. And we've heard from the fantastic talk this morning by Magdalena how important it is to look at the single cell level and to really understand the biology of these mechanisms. And so this is what our group does. So we're trying to really understand who these immune cells are that participate in this immune response to pregnancy, and what they do, how they behave. And we measure their signaling pathways that are at the protein level, that are really the building blocks of these immune cells' response. So how do we do this? So we use this technology called CyTOF, which I want to say in the last 5 years has really revolutionized how we look and we visualize and we understand the immune system. CyTOF has introduced a new discipline of high-dimensional flow cytometry. It was first invented by Scott Tanner in University of Toronto, but really developed here at Stanford by Sean Bandel in Gary Nolan's lab. And the way it works is very similar to traditional flow cytometry, if you know flow cytometry. Just like fluorescence flow cytometry, it's a single cell technology. So every immune cell present in a blood sample will be analyzed as a single statistical event. But unlike flow cytometry or traditional flow cytometers, now we can measure many, many, many different parameters per single cell. Very easily, in a routine analysis, we measure as many as 50 proteomic parameters per immune cell subset. And we do this by using antibodies that are conjugated to metals. And we use the dynamic range of mass spectrometry to measure many different antibodies at the same time. And there's two parts in the analysis. We can measure surface markers and therefore precisely phenotype every one of these immune cells in a blood sample. Okay, we can use, for example, 20 surface markers and phenotype all immune cells in a blood sample. But at the same time, by using intracellular antibodies, we can go inside the cell and measure the activity level of various signaling proteins, transcription factors, kinases, etc. So we can use another 20 or 30 parameters to now understand what is the behavior of this particular cell that we're looking at. So now we have 50 parameters to play with to visualize the immune system with really unprecedented resolution. And we can use, for example, unsupervised clustering algorithms to map out the immune system of a patient in a patient sample. This map is made of different clusters that are made of many different cells that have the similar phenotype from the neighboring cluster, but very different than another branch in this map. And you recognize the major groups of immune cells. Your CD4 T cells, your CD8 T cells, your innate cells are present, but you see that with this resolution, we can subset these major populations into very discrete clusters of cells that have different phenotype. And this is just the first part of the analysis. Then for every one of these cell subset, we can ask, well, how is it behaving? And importantly, this is at the proteomic level. Okay, we're looking at phosphorylation changes. We can look at abundance changes, total protein, but this is really at the protein level. We can look at as many as 10, 20 functional markers as well in every cell subset. And so we've used this technology to really try to understand how does the immune system adapt to pregnancy, how does it evolve? And the end goal here is to answer the question, is there an immunological clock of pregnancy? Is there a set of immune features or immune mechanisms that track pregnancy and that will determine precisely when the time of labor has come. This is the end goal. But before answering this larger question, we wanted to answer maybe a simpler question, which has to do with when does the timer start? So for the remaining part of the talk, I want to share with you the results of a cross-sectional study where we enrolled women that were non-pregnant but had either a history of term labor or preterm labor. And so therefore, the preterm group were at high risk of developing preterm at the next pregnancy. And we asked the question whether in this at baseline, in the non-pregnant state, can we distinguish differences in their immune system? Or are they at baseline different? They're the immune states of these two patients group. This is the summary slide of the results of this study. The first part of the analysis. It's important to understand that here in this slide, this represents a data set that contains at least 70 million data points per patient, and altogether it's over 1.5 billion data points. And so you see that we could build these maps of these two patients, the immune system of these two patients group, and then ask, looking at every one of these immune cell subset, ask whether their function and their response to various extracellular stimulation were different from one patient group to the other. And the answer was yes. And it was biologically interesting. What we were finding here is that the ability of subsets of innate immune cells in the classical monocyte subgroup compartment here had an exacerbated response when stimulated with a toll-like receptor 4 agonist, and TLR4 is one of the major signaling pathways in response to extracellular pathogens. And so the answer to our question was yes, we can differentiate, we can pick up on immune features that are different between these two patients group. That was satisfying. However, if you think about it, this analysis was fairly simplistic as a first pass, because what we were doing here is we were taking every one of these immune cell clusters individually and asking, okay, is this one different between one patient group or the other, and then looking at the other one, et cetera, et cetera. However, the immune system is not that simple, and all these immune cell subsets don't act in isolation. They correlate with each other, they talk to each other, they communicate with each other, and these signaling pathways move together or antagonistically with each other, and they talk to the environment as well. And so this represents the same data sets with all the correlation networks or the correlation between those immune features highlighted in gray here. A much more complex data set. And so in the second part of the analysis, we wanted to see if we could harness this amazing information contained in all these correlation in order to see if we could differentiate between these two patients group and see, with better predictive value, if we could now see what type of immune features was predictive or associated with our clinical outcome, preterm history here. And so this is a complex statistical problem because we're making now many different comparisons, and we only have a few patients to play with. So fortunately, we work with Nima Gaipur is our bioinformatician here and a co-investigator on this study. And he worked in close collaboration with Rob Tibshirani and adapted one of his penalized linear regression model called ElasticNet. And what that did was to take this information, this correlation information, and distill it down to a few sets of immune features that formed a network that, when measured together, was strongly associated with having a history of preterm birth. And the reason why that is exciting is this is a cross-validated result. The effect size is very strong. The p-value 10 to the minus 7. So really comforting us into the statistical soundness of the results, and also encouraging us for future studies, validation studies, and future longitudinal studies. So I want to finish with this slide here to tell you a little bit where we're going with this. So here we are. We have a 50-dimensional mass cytometry assay We can detect various immune features that form networks of immune networks of correlated features. We can ask whether these networks make biological sense by going to the bench and stimulate them and interfere with them in vitro. And that's interesting. But really where we want to be as soon as possible is there. We'd like to build a diagnostic test that can be used to predict whether a woman, prospectively, whether a woman will have what is at risk to deliver preterm, so that adequate care can be delivered to her. And so there are two major milestones towards this goal. One has to do with scalability, and the other one with reducing the dimensions. Sorry, I should go back to this. So scalability is actually something that is one of the milestones that I think we've already figured out, because although this is a complex assay where you need specialized facility to accomplish, it's performed on a simple blood draw that can be, and it's performed on a whole blood system that can be fixed and banked and frozen in a freezer and then shipped from anywhere around the world towards an adequate facility. And we already communicate, we already collaborate with many centers internationally that ship us their blood samples. So I think we can scale up this assay fairly easily. Now, reducing the dimensions is another problem, and we're working really hard on this, because obviously at the end of the day, we want an assay that can be tailored to any kind of clinical flow cytometer, which typically have 4, 5, maybe 6 parameters. And so on our way to answering this second problem, Nima just wrote an algorithm called Gate Finder. And what this thing does is really once you know what immune features you have identified, once you use your 50 parameters to identify the predictive immune features, then you can determine which one of these, or the minimum number of parameters that are necessary to capture 99% of the information contained in this immune network. And that way, using these parameters, you can get as almost all the information, the predictive information necessary to predict your clinical outcome. And therefore, once you have these 4 or 5 parameters, you can then use them in a clinical platform. And so with this, I want to thank first the organizers of ChildX, and thank you for inviting me. And then obviously this is a collaborative work with a very interdisciplinary group of people. I want to thank the people in our lab, so Martin, Nima, and then all the other guys, Ed, Martha, Leslie, Hope, and Quentin, that have worked on this projects. And then David Stevenson's group and the March of Dimes and Center for Prematurity here at Stanford, which is an amazing group to be part with. And then, of course, Gary Nolan's lab, my former mentor, and I still work with a lot of people in this lab as well. Gabby and Rob particularly contributed to the work significantly. Thank you, and I'll take any questions.
Thank you very much. Wonderful talk. We have time for a couple of questions.
So that's the next step. Obviously, there's progesterone, and we know that progesterone has immune modulatory properties. So that could be one intervention to see how does this affect the network that we have at play, and see if we can, once you have a predictive test, if you can target women that are at high risk with this. But there's room to work.
Other questions? Repeat the question, perhaps, or summarize.
So let me see if I understand the question. So the predictors are based on comparing a preterm group with a history of preterm group, and you're asking if you were to take 2 groups of women that don't have this history of preterm, how does the predictor look? It's difficult to answer, because the way the learning algorithm works is by focusing on these differences between the preterm history and the history of normal terms. So this is how those features are found. But I think it opens up a different question, which we're actively working on. Is these are features that have been found at baseline in a non-pregnant state that can be either a result or a cause of the following preterm birth. And so really to answer this, we need to go in longitudinal studies, and that's what we're doing. So we have cohorts of women that we're following longitudinally through pregnancy, and some of them have a history of preterm birth, and we want to see if the same immune features appear and can predict now first their history, and then also if they're going to go ahead and deliver preterm at the same time. The caveat here is that it's very hard to have a baseline sample in the non-pregnant state. There'd be some ethical issues here. And so the way we address this is we take a blood sample postpartum as our baseline, once 6 weeks to several months postpartum as our non-pregnant baseline.
Wonderful. Thank you very much. Oh, was there another question?
Yeah.
Oh, yes, go ahead.
Compared to those mothers who always have overdue, the pregnancies go.
That's really important. So it goes back to the idea of an immunological clock. Is the preterm phenotype a continuous phenotype from delivering preterm and delivering postterm? That'd be a really good biological control whether the mechanisms that we're looking at are actually the same and affected differently in postterm pregnancies. And so we're looking this in a fairly small cohort of longitudinal patients, but it'd always be great to move on and have larger cohorts.
Okay, I'll have to move on. Thank you so much.
Thank you. No, thank you.
Thank you.